Right, so this is a bit of an older story from before that Chip wanted us to bring up again and to discuss the pathogenesis of DATA2 as well as some of the basic mechanism that we can see from some of our patients. These are disclosures. And I want to talk a little more detail about the first case of DATA2 at Boston Children's. This is certainly not the first case that went through Boston because we now know there are many cases before this one that we just didn't know at the time. 2-year-old girl came to me with intermittent fever, skin rash that you can see on the two pictures. So she has two types of rash, this erythematous papules, and then she has livedo in the background. She has chronic anemia, hemoglobin was as low as 7, elevated inflammatory markers, and most remarkably, she was not gaining weight for almost a whole year. We thought she had CAPS, right? The rash looks sort of like that, a lot of inflammation. We got lots of different consults, never found an infection. And this was at the time where there was no Invitae panel or something that's that quick, right? We didn't have that option. So we did stand up genetic panel. We started her on IL-1 antagonists, both anakinra and canakinumab in sequence, didn't really help. We did some cytokine panel. Her IL-6 was quite high, as well as her CRP. So we thought about, well, why don't we try tocilizumab, which fixed the numbers, right? She was also doing better in terms of appetite and has more energy, and then her anemia actually improved significantly. But after a few doses, she came in one night with progressive lethargy and vomiting, and CT scans show this on the top picture there. So a large hemorrhagic stroke, went to craniectomy right away, and while she was in ICU, a few days later, she had another stroke, now on the opposite side, bleeding into the ventricles, see in the bottom picture there. So we're all still waiting on the sequencing, of course. Then it was Mike and Ivona that came to the rescue. So we got ADA2 activity through Mike's lab, and we can show that the patient had deficient activity, shown on the graph here. Mother had partial deficiency, so sensitive a carrier. And from Ivona's lab, we can found two mutations. One is a missense mutation of T129P, and then there's another deletion involving some of the latter exons. When we sequence the cDNA, we can only find the missense mutation, meaning that the deletion probably led to RNA instability, and we don't see that from the transcript level. Okay. So T129P was a new variant at the time, so I want to understand why does this cause ADA2 deficiency, or is it the cause? So we cloned this mutation into a plasmid and transfected into 293 cells. And using different concentration of plasmid, you can see wild-type cells will release different amounts of ADA2 in terms of activity on the supernatant. And in the mutant construct, at any concentration you transfect, we don't see any activity on the outside. When we looked at inside a cell using antibody to the MICTAC ADA2, we can see a protein expression, right, even for the mutation, except that it's a little bit smaller. So that's a little strange, right? You change one amino acid, and you have a shift in the molecular weight of the protein. So what's happening there? So we thought maybe there's some type of post-translational modification. And this is from me sitting through the first DATA2 meeting, actually. Somebody showed a slide, right, and showed these glycosylation sites, which sits exactly where the mutation is. So 129 is part of this sequence of N, X, and S or T, right? So you have asparagine, anything in between, a serine and a threonine. These constitute a glycosylation site where the cell puts a sugar residue onto the protein that will allow it to traffic to the appropriate place. So we can do this experiment to show that wild-type ADA2, you can detect it inside the cell, you can get it outside the cell. If you change the T to the P at 129, you lose the expression, you lose secretion, and the expression inside the cell is a little bit smaller. You reverse that mutation in the same construct, you can get it back. And we can also make a mutation in site 127, which is also part of this glycosylation sequence, and get exactly the same result. So this is the first demonstration showing that this is likely to be a glycosylation defect causing this patient mutation. We can also do this using tunicamycin, which is a blocker of glycosylation with both wild type and some of the mutant proteins that you can see by blocking glycosylation, you can drop the expression or the secretion of the wild type protein by quite a bit. And this is a separate experiment also showing that in the control wild type ADA2, if you add tunicamycin, you can drop the molecular weight also. So very consistent with where we demonstrated that glycosylation as the explanation to the molecular weight of this mutant protein. So glycosylation turns out a quite important process. Right? It's important for protein folding, trafficking, stability, and function. And so what's going on with this protein? So with this confocal microscopy, it showed that if you express wild-type ADA2 in 293 cells, you can get colocalization around the nucleus in a distribution that's what's described as Golgi localization. Okay? In green is a ER protein, so you don't see, in terms of the overlapping, you don't see a ton of localization to the ER. In the mutation, T129P, we lose that Golgi distribution, and everything now co-localizes to the ER, right? So you can see all the entire cells in yellow. You can see the same thing with N127 substitution that also causes a glycosylation defect. Again, you can see a complete overlap in yellow. And we also did Giantin, which is a Golgi staining, to show that this overlap indeed is the Golgi, and you don't see Golgi co-staining with the 129P mutant. So looking at the protein, there are actually 3 other glycosylation sites. So predicted here at amino acids 127, 174, 185, and 378 as the beginning of the NXST sequence. So these things are highly conserved through evolution, with the exception of the 185, right, through many different species. And when we knock out or make the change in these other sites, they all shift down their molecular weight by a little bit, showing that they are indeed glycosylated, right, at least from an in vitro standpoint. But the 185 site for glycosylation did not affect secretion. So the protein still gets made, it still gets secreted. That's different from the other 3. So 127 we talked about already, and 174 and 370 also are very similarly important for trafficking of this protein. We did very similar experiments in colocalizations, and to show that, you see the Golgi staining with the wild-type protein. You don't see it with the N174. You see it again with the 185, which is not required for secretion, and you see it really ER pattern again with the 378. So this experiment will show that among the other 3 glycosylation sites, 185 was not required for secretion of the ADA2 into the extracellular space. We can also demonstrate that some of these mutants, even though they're not getting secreted outside, there's still a little bit. And if you look at the little bit of protein that's outside, you normalize it to the amount of protein in terms of ADA2 activity, they also actually have some ADA2 activity. So the glycosylation mutants actually didn't affect ADA2 enzymatic function very much. I think the defect is much more in terms of the localization. This is just a plot to show the ADA2 activity, showing that wild type and 185, which did not affect the secretion, have normal ADA2 activity, versus the other one had reduction, but still a little bit detectable activity. And then going to the NOMAD database, we asked, well, what other mutations are that could affect glycosylation? And there are a couple of gain-of-function variants in the population where instead of having a loss of function, right, these are mutations that could give you a new glycosylation site. And it's just a curiosity experiment to what happens to those. And you can see here, this one leads to an increase in molecular weight, right? So now consistent with now getting an extra glycosylation site. This one has sort of a partial effect, a little bit of a shift. In both cases, you do have also loss of function in terms of ADA2 secretion in these constructs. So to conclude, we found that T129P in this patient was a pathogenic mutation that disrupted proper N-glycosylation of ADA2. You do need proper glycosylation in 3 sites for proper trafficking of ADA2 from the ER to the Golgi to the outside. We did not study the lysosome in that particular experiment. It will be very interesting to consider that. So we can show that both loss of function and gain of function glycosylation mutants can profoundly affect ADA2 secretion. And this is also important in thinking about recombinant ADA2, because I do think proper glycosylation is really important for the stability and generation of this therapy. So thank you for your time.